How cells differ. Let's talk about epigenetics now. What is epigenetics? Have you heard of this term before? Breaking the word apart, epi means above, and genetics obviously means genetics. And so technically, epigenetics means everything above or surrounding genetics. So the genetic code contains a lot of information, but there's a lot of proteins and molecules that help us read the code and implement it. And so the discipline of epigenetics mostly refers to proteins that help structure DNA and molecular marks that actually change the nucleotides and make chemical changes to the nucleotides that help annotate the genetic sequence. And so in general, this is what the discipline of epigenetics is looking at. Let's have a look at how epigenetics helps in the formation of our highly condensed chromosomes. We'll start with DNA, the double helix as we're used to seeing it. And we can see that it's wrapped around these protein complexes, almost like beads on a string. And if we look closely, we can see that these beads are made of these protein complexes. So the whole entire bead with the proteins is called a nucleosome, and each of these small proteins is called a histone. There are 8 histones together, and DNA wrapped around it make up the nucleosome. And so here we have a string of nucleosomes, and these nucleosomes get tighter and tighter, very tightly wound, forming this tighter coil of a solenoid. And this very tight solenoid gets wrapped up again into the supercoiled structure we know as the chromosome. And so we've seen how these proteins help form a chromosome, but this is not everything that epigenetics is concerned with, just an introduction. And so we can see that there's a lot of proteins that are involved. Also, methylated cytosines are the molecular marks we talked about earlier. Here we can see that the cytosines in the sequence actually have modifications on them. So going back to the proteins, what is important for you to understand is that histones are proteins that bind to DNA, and histones wound together with DNA form a nucleosome. And it's these nucleosomes that help wind the DNA into more complex structures. Methylated cytosines are an actual modification that occurred to the cytosine nucleotide. Here we can see a regular, this is what the cytosine nitrogenic base looks like normally, and this carbon gets a methyl group added, and so this is a methylated cytosine. And this methylation only happens to cytosines. This is a frequent epigenetic mark that contains a lot of information, but we'll talk a bit more about this later. But in summary, histones and nucleosomes are the proteins, the major players in epigenetics, and the molecular marks are these methylated cytosines. To have a look at what I just showed you with chromosome folding, let's see about how these proteins bind to DNA in real time. There's a really good video that showcases this. You can click on this link or just watch the next video in the playlist.